Assalamu alaikum class. I hope you are all fine. Here is the course content again. We have so far covered the classes of protein tertiary structure in our previous week, and now we will move on to the enzymes. The enzymes. The enzymes are the reaction catalyst of biological systems. They are the most remarkable and highly specialized proteins. Enzymes have extraordinary catalytic power, often far greater than that of synthetic or inorganic catalysts. They have a high degree of specificity for their substrate. They accelerate chemical reactions tremendously and they function in aqueous solutions under very mild conditions of temperature and pH. Only few non-biological catalysts have all these properties. In this series of lectures, we will be studying about enzyme chemistry, their classification, mechanism of action. We will also be studying about enzyme kinetics briefly and how the enzymes are inhibited, activated, and also about their specific nature. First of all, we have enzyme chemistry. Enzymes are central to every biochemical process. Acting in organized sequences, they catalyze the hundreds of stepwise reactions that degrade nutrient molecules, conserve, and thus transform chemical energy, and make biological macromolecules from simple precursor. With the exception of a small group of catalytic RNA molecules, all enzymes are proteins. Their catalytic activity depends on the integrity of their native protein conformation. If an enzyme is denatured or dissociated into its subunits, catalytic activity is usually lost. If an enzyme is broken down into its component amino acids, its catalytic activity is always destroyed. Thus, the primary, secondary, tertiary, and quaternary structure of proteins, enzymes, are essential to their catalytic activity. Some enzymes require no chemical groups for their activity other than their amino acid residue. Others require an additional chemical component called a cofactor, either one or more inorganic ions. Some enzymes require both a coenzyme and one or more metal ions for their activity. A coenzyme or a metal ion that is very tightly or even covalently bound to the enzyme protein is called a prosthetic group. A complete catalytically active enzyme together with its bound coenzyme or a metal ion is called a holoenzyme, as you can see in this figure on the left and the right side. The protein part of such an enzyme is called an apoenzyme or apoprotein. Coenzymes act as a transient carrier of specific functional group. Most are derived from vitamins, organic nutrients in small amounts which are required in the diet. In the figure you can see the cofactors. The cofactor is the non-protein molecule which carries out chemical reactions that cannot be performed by standard 20 amino acids. Cofactors are of two types: organic cofactors and inorganic cofactors. Inorganic cofactors are metal ions such as iron, magnesium, manganese, or zinc, while organic cofactors are complex organic or metallo-organic molecules. Here we have cofactors and their classification. As you can see, cofactors can be organic or inorganic. The ones which are organic have a prosthetic group which is tightly bound with the help of a covalent bond to the organic cofactor. Like you can see the FAD or flavin cofactor in the figure on the right side. The heme group of hemoglobin and myoglobin are also the prosthetic groups that we discussed in the globular protein series. Then we have the other class of organic cofactors called the coenzymes. The coenzyme is loosely bound organic cofactor. For example, the NAD cofactor. This NAD cofactor is helped with the help of a non-covalent interaction, so it is loosely bound. Now the structure of an enzyme. The distinguishing feature of an enzyme catalyzed reaction is that it takes place within the confines of a pocket on the enzyme called the active site. As you can see in this figure of an enzyme on the right side of the slide, the blue and red color part is the active site of this enzyme. Active site generally occupies less than 5% of the total surface area of an enzyme. And as it has a specific shape due to its tertiary structure of the protein itself, that's why if we change the shape of a protein, the shape of this active site will also change and it will eventually affect its activity. Active site can be further divided into binding site and a catalytic site. The binding site chooses the substrate and bind it to the active site, while the catalytic site performs the catalytic action of the enzyme. The molecule that is bound in the active site and is acted upon by the enzyme is called the substrate. Look at the cartoon figure at the bottom of this slide. The pizza shaped guy, blue in color, is the substrate, while the yellow one is the enzyme. The surface of the active site is lined with amino acid residues with substituent groups that bind the substrate and catalyze its chemical transformation. The green line that you see in the mouth of the yellow guy, that's those amino acid residues. Often the active site encloses a substrate, sequestering it completely from the solution. Let's have a very brief look at the site of enzyme synthesis and enzyme types, depending whether they are active inside the cell or outside the cell. You all know how translation takes place and how proteins are synthesized. Since enzymes are made up of proteins, they are synthesized by ribosomes and they act as biological catalysts. And you all know that these proteins, or we should say here the enzymes, are produced inside the ribosomes, just like all of the proteins. You know, the most of the ribosomes are located on the rough endoplasmic reticulum. The ribosomes are located inside the cell, so these are the organelles, or we can say that these are one of the cellular organelles. So let's look at the differences between the intracellular and extracellular enzymes in a general way, and then we will see what are the differences in their biosynthesis within the cell. Not only their biosynthesis, but also their sites of action. Both the extracellular and intracellular enzymes are proteins synthesized in the same way like any other protein, but the difference is how and where they are being produced and they are being used up. So the first intercellular enzymes, every cell produces its own enzyme for doing all the intercellular activities like the cellular respiration, oxidative phosphorylation, which are going on inside the mitochondria. These enzymes are produced by the cells and they are being used up by the same cell. So these are intracellular enzymes. While in case of extracellular enzymes, it's different. In the case of extracellular enzyme, when the enzyme is produced by the cell, it is exported out or secreted out. It is not used by the cell itself. When the enzyme is secreted out into the extracellular space, it will be used by other cells. So that the enzymes that are used by the other cells rather than by the one which is producing it, these are called as the extracellular enzymes. Now, with respect to the differences in the biosynthesis and their transport, the two enzyme categories, I'm going to explain them. And to explain them in a better way, I'll be using a laser pointer as well. Now, since we all know that enzymes are produced, they are proteins and they're produced exactly in the same way as all other proteins are. So the enzymes in both cases, whether they are being produced intracellularly or extracellularly, are produced in the same way. As you can see here, and in the ribosomes, now the ribosomes are the red dots that are attached on the rough endoplasmic reticulum. all the messenger RNA for the enzyme would be translated here. After being translated, the polypeptide or proteins that are formed, these proteins are the enzymes. After their synthesis, inside the endoplasmic reticulum, they are going to bud off from the endoplasmic reticulum. Actually, when the proteins are heading to the Golgi body from the endoplasmic reticulum, they travel inside a vesicle. These vesicles, since they are transporting the proteins or the enzyme from the endoplasmic reticulum to the Golgi body, these are called as transport vesicles. Once these transport vesicles arrive at the Golgi body, they are going to fuse with it. These enzymes, when inside the Golgi body, they keep on moving and traveling until they reach the other end of the Golgi body. Once they reach the other end, they are again going to bud off from the Golgi body, but this time the proteins will be released directly into the cytoplasm. This is how the intracellular enzymes are produced and then used up by the cell, like they are being produced and then after budding off from the Golgi body, being used up directly within the cytosol or the cytoplasm. Now when we look at the extracellular enzymes, how they are produced in the same way. The only difference is at the site of the Golgi bodies. Instead of budding off and being released from the Golgi body directly into the cytosol, the proteins, when they are released from the Golgi body, they are again retained within a vesicle. This vesicle is exactly like the transport vesicle, but now this is called a secretory vesicle. These secretory vesicles move towards the plasma membrane instead of being used up within the cell. And when they fuse with the plasma membrane, they release all the proteins which are stored inside them, which are the enzymes. So the enzymes in this way are released out of the cell which is manufacturing it. These enzymes, now called the extracellular enzymes, are used up by the cells which are surrounding the cell which is actually synthesizing that.